Okay, can you see the screen now, male reproductive system?
Yeah.
Okay, yeah. So now we are going to start chapter 4, the male reproductive system. We are going to just start a few parts of this chapter. This is a relatively longer chapter than the other ones. So it could take 2 or 3 weeks to be done with this chapter. So first of all, what are the different parts of the male reproductive system? The most important of all the parts, which is the testes, is the organ that will produce the sperms, but also it will be producing the steroids. So it's the testes are important for spermatogenesis as well as for steroidogenesis. This is because the Leydig cells, they secrete the testosterone, which is a very important hormone for the development of the male. In addition to the testes, of course, so you have the penis, you have the testes, and a very important thing that is not shown here is the epididymis. So this one here is the epididymis, and the epididymis is very, very important as part of the male genital tract because the epididymis is responsible for the maturation of the sperms. لأنه sperms وقتها يتركوا the testes when they leave, so they are formed inside of the testes, and particularly they are formed inside the seminiferous tubules. So when they leave the seminiferous tubules to go outside of the body, the purpose is to ejaculate them later on. So when they leave these sperms, when they leave the testes, they are not mature. They are partially mature, but they are not 100%. So the maturation continues at the level of the epidermis. So this is very, very important. Now, so the epidermis, they ensure that the maturation of sperms, so in the epidermis, and then you have the genital excurrent ducts that leads to the transport of the sperms. So عندنا the ductus deferens. اسمها الثاني إذا مرقت بالكتاب ما بعرف إذا it's written ductus أو vas. Okay, so ductus deferens or vas deferens, it's the same. There is the ejaculatory duct, and of course the last stage is the urethra. So all of these are there to transport the sperms to outside of the body. Moreover, very important things are the accessory glands. The accessory glands, لولاهن, the sperm that will be ejaculated will not be a functional sperm. These accessory glands will be secreted, whether we are talking about the seminal vesicles, there are two, or the prostate gland وحدة, and the bulbourethral glands أو بنقلها اسمها الثاني Cowper glands. So it's the same. Cowper gland وبالبيوثرا is the same gland. So عندنا اثنين. So اثنين باثنين بوحدة prostate وحدة. All of these accessory glands are very important because they will be secreting, حنشوفها in details in the next week or the week after. They will secrete molecules and fluids that we call the seminal fluid that will be mixed with the sperm cells. لما بنقول السائل المنوي, it contains it's the semen in English. So semen, سائل منوي, it contains the sperms اللي هو الحيوان المنوي and the secretions. Okay, so the seminal fluid is the mixing of the different compounds and molecules that will help the sperm resist and be able to be stable till it reaches the female reproductive system in case there is copulation. Okay, so these are very important as accessory glands. How are the testes organized? We mentioned in the previous chapter that these testes should be descended. لازم ينزلوا through the abdominal cavity to outside of the abdominal cavity to be located in the scrotum. Why is this important? I mentioned no, they should be located at a temperature that is 2 to 4 degrees lower than the body temperature, and this is very important and crucial for spermatogenesis to occur properly. And of course, I mentioned also that these on the testes, on this location, on the posterior superior region of each testes, there is the epididymis that is located. And the functions of sperms is the spermatogenesis, production of sperms, as well as the synthesis of steroid hormones, which we call the steroidogenesis. What about the anatomy of the testes? So what about the different parts of the histology بالأحرى of these different, what are the different tissues that makes these organs? Now, first of all, the important layer is the thick, dense, fibrous connective tissue that we call the tunica albuginea. Now, the tunica albuginea, which is pointed here in this figure, is made of a dense, fibrous connective tissue, and it's like a capsule that covers each of the testes. What is the importance of this tissue? This tissue is crossed by testicular vessels and consists also of collagen fibers and smooth muscle cells. So it's very important, the presence of these smooth muscle cells. This will allow this tissue, the tunica albuginea, to contract spontaneously and rhythmically, and this contraction will help the sperm move out of the testes till they reach to the epidermis. لأنه أول مرحلة they are produced in the seminiferous tubules. They go into the lumen, and then they are transported through the ducts till they reach to the epididymis. In order for these sperms to move من التستيز ليوصلوا على epididymis, and of course in order for the fluids that are present with these sperms, so sperms with testicular fluid, to be propelled out of the testes, they need contraction, and this is given and provided by the smooth muscle cells that are part of this tunica albuginea. Moreover, this tunica albuginea is innervated by adrenergic terminations, which means that it works on adrenaline and noradrenaline. Okay, so it can respond to adrenaline and noradrenaline. And this is very important because it will contribute to the steroidogenesis and to the spermatogenesis, since it responds to adrenaline and noradrenaline. Now, at the upper edge of the testes, this tunica albuginea will thicken here, it's thickening, and will sink deeply to a region here that we call the body of Highmore. What is the body of Highmore? شو مفروض تتخيلوا؟ It's like a roundabout مثل رون-بوان. اوكي مثل رون-بوان، اعتبروا في طرقات بيوصلوا على رون-بوان مستديرة. This place here is called the body of Highmore. It's like a tissue or a place inside of the testes that is perforated by vessels and the channels. لاحظوا the channels, they are very clear. These channels we call them the rete testes. وهول الchannel هن continuous مع seminiferous tubules. هول, these are the seminiferous tubules. They will all of these seminiferous tubules, they will all converge toward this body of Highmore. So this body of Highmore, which is made of rete testes, are channels that are continuously connecting with these seminiferous tubules. هلا بقلب we said that we have tunica albuginea, we have the mediastinum or the body of Highmore. Now between inside of this part, the cortical part of the testes, بينوجد another tissue اللي هو a connective tissue but not a dense one. التونيكا البوجينيا قلنا انه it's a dense connective tissue. بينوجد a loose and areolar connective tissue, which is here. It will be located in this place. By being located, it will be like limiting or dividing this part of the testes into compartment that we call the lobules. هيدول loose connective tissue بنسميهم septa. They will be dividing all of these testes into lobules, and they will divide the testes into 200 to 300 lobules. فإذا شو هن السبتا؟ كيف انعملوا؟ هن عبارة عن loose connective tissue, loose areolar.
كثير عم بيروح الصوت، عم بيقطش كثير من عندي.
Okay, one minute. Okay, is it better now? Can you hear me better?
Yes.
Okay, so what I was you can still see my shared screen, right?
Yes.
Okay. So اللي عم قوله انه عندي two types of connective tissues within inside of the testes اللي هن directly in contact مع اللي هن اقرب شيء للseminiferous tubules. عندي the dense connective tissue التونيكا البوجينيا، وعندي the loose connective tissue. التونيكا البوجينيا هي محاوطة كل الseminiferous tubules. ببعض المحلات بينوجد loose connective tissues. These loose connective tissue بيقسموا التونيكا البوجينيا into lobules. Okay, so they will limited or divided into lobules, and inside of these lobules there are the seminiferous tubules. Now here you can see in this figure the seminiferous tubule. So كل وحدة من هول is seminiferous tubule. كل lobule قلنا 200 ل 300 lobule. They contain 1 to 4 highly coiled seminiferous tubules. وقلنا انه they become seminiferous tubules after puberty لانه they acquire lumen. بصيروا مفرغين، بصير تيوب، يعني هو هيدا من جوا فاضي. Okay, these seminiferous tubules will converge toward the body of Hymor محل ما موجودين الريتي تاستيز. شو اللي بيوصلهم بالريتي تاستيز؟ لاحظوا هون في محل straight. فإذا عندي seminiferous tubules highly coiled بيرجع بيجلس. This part that is straight, we call it the straight tubules, is the one that connect the seminiferous tubules to the straight tubules to the rete testes at the level of the body of Hymor. فإذا لما ينصنعوا، لما يخلصوا يصير في spermatogenesis and sperms are released in the lumen of seminiferous tubules, these sperms will go through the straight tubules, through the rete testes, then to the efferent ductules to reach to the epididymis. Okay, this is the way it will be transported. What about the cross-section of a seminiferous tubule? Hey, cross-section of seminiferous tubule. كل seminiferous tubule عنده صار lumen لأنه قلنا إنه after puberty it has acquired this lumen, so it's centered. So the lumen is there, and it is limited. It is surrounded by Sertoli cells مع the germ cells all over it. وكمان it is limited بين seminiferous tubule والحدو. It is limited by a connective tissue كمان. Now this connective tissue is made of, it's called the lamina propria. هالlamina propria من شو معمولة؟ It's made first basal lamina اللي هي the basement membrane أو basal lamina, and a group of collagen fibers, fibroblasts, and some contractile cells بنسميهم peritubular cells موجودين كلهم بالlamina propria. Lamina propria اسمها, we can call it also the proper membrane. So the proper membrane أو lamina propria هي عبارة عن basal membrane أو basal lamina اللي هي directly in contact مع seminiferous tubule، وتحتها في layers of collagen fibers and layers of peritubular cells أو بنقول لهم the myocytes, which we will be talking about later in this chapter, and fibroblasts also are present inside of it. And this is here the connective tissue, and here is the lumen. So هون إذا بدي آخد هاي مثل هاي الصورة كأنه عاملة cross section in this way. Okay, so I have the lumen here and there, and I have here the basal membrane, and here I have the basal membrane. Now, inside of the seminiferous tubule, I have the sperm cells, I have the spermatogonial stem cells, I have the primary spermatocytes, secondary spermatocytes, spermatid. So all the different levels and stages that the germ cells are in are present here. I have a big large Sertoli cell. هاي شايفينها لونها هون purple. هيدا هي السيرتولي cell. It goes from the basal compartment till the lumen. وحاضنة قلنا انه هي mother cell لانه هي حاضنة all the different stages of sperm cells of germ cells من السبرماتوغونيا للبرايمري سبرماتوسايت للسكندري سبرماتوسايت للسبرماتيد حتى نوصل للسبرماتوزوا that will be released in the lumen of the seminiferous tubule. بينما Leydig cells مش موجودين بقلب السمنيفرس tubule بيكون موجودين برا. It's in the interstitial media between the seminiferous tubule. هون 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 بيكون في Leydig cells وبيكونوا موجودين هال-Leydig cells in contact مع blood capillaries that are present in the vascular compartment outside of the seminiferous tubule. What about the peritubular cells? These peritubular cells اللي هن part of the lamina propria هن similar to smooth muscle cells, هن contractile cells, and they are very important because they participate in the evacuation of sperms from the seminiferous tubule. بيكون في 3 to 5 layers of these myoid cells, again with collagen, fibroblast, وعند الممbrane الأساسية is the basal lamina. What about the types of cells that makes the seminiferous tubule? We have two types of cells. Inside of the seminiferous tubule, we have the spermatogenic cells, including spermatogonia, primary spermatocyte, secondary spermatocyte, spermatid. All of these are spermatogenic cells. وفي السيرتولي cell اللي قلنا it's a support for spermatogenesis. We call it the sustenticular cell أو the nursing cell أو the supporting cell. In the interstitial fluid أو space, there is a connective tissue بقلبه بينوجد Leydig cells, and these Leydig cells are present along with lymphatic system and blood capillaries, and these are cells that have endocrine activity. It has secretory activity. It's the producer of testosterone. It is present in the connective tissue between the seminiferous tubule and not inside of the seminiferous tubule. This is another figure that is showing all. So Leydig cell برا, Sertoli cell جوا. Leydig cell secretes the androgens. So androgens are secreted by Leydig cell. بينما spermatogenesis بتصير بقلب الseminiferous tubule. And these cells are in close contact مع هيدا big cell that you can see, which is the Sertoli cell. Thus, to recapitulate on this and these cells, so we have Sertoli cells. It's a columnar cell going from the basal compartment to the lumen. This cell is a supporting cell. This cell does not proliferate after puberty, and this cell, very interestingly, we will see this later on, عندها processes. لاحظوا انه هيدا السيرتولي cell وهي السيرتولي cell كأنه ماسكين ايدين بعض, as if they are holding the hands of each others. And this is very important لأنه بيعملوا limitation, بيقسموا لي seminiferous tubule into a basal compartment and an abdominal compartment, and this will be discussed later. ليه هيك بيعملوا؟ كمان عندهم apical extensions. كمان لاحظوا هون they have apical extensions. Okay, so these are very important lateral and apical processes of these Sertoli cells. بينما Leydig cells موجودين برا, so they are interstitial cells and they synthesize androgens. بما انه they synthesize androgens اللي هن steroids, يعني عندهم production of lipids, يعني عندهم extensive smooth endoplasmic reticulum. بينما spermatogenic cells اللي هن هودي كلهم كل المراحل they are replicating, some are differentiating, and we go from the least differentiated one بتكون toward the periphery spermatogonia to the most differentiated one اللي هن sperm cells toward the lumen. Now, the last part I want to talk about today is the, before I stop, is the beginning of spermatogenesis. Now, spermatogenesis is the process that will allow us to produce sperms from stem cells. Remember when I talked about the primordial germ cells, primordial germ cells becomes prospermatogonia. Now, this prospermatogonia needs to differentiate into stem cells, and these stem cells will be producing these spermatogonia صار اسمهم اللي هن spermatogonial stem cells will be producing primary spermatocyte, secondary spermatocyte, spermatid till we reach to the sperms. All of this mechanism, all of these steps takes 74 days, and every 74 days 100 millions of sperms are produced. يعني مش كل يعني مثلا إذا بلشنا اليوم بعد 74 يوم we produce. إذا بلشنا بكرة بعد 74 يوم we produce. So every 74 days there will be a batch of 100 millions of sperms that will be produced. The spermatogenesis occurs in 3 steps. The first one is mitosis. The second one is meiosis, and we have the differentiation or the spermiogenesis. Mitosis plus meiosis plus spermiogenesis takes 74 days. So we have, for example, mitosis 27 days. Then you have the 24 days for meiosis. Then you have 23 days for cytodifferentiation. So all of this will lead to the formation of the mature sperm after 74 days. And this spermatogenesis will start at puberty. ليش؟ لأنه the gonads are now responsive to the gonadotrophin hormone اللي هن ال FSH وال LH. ببلشوا يتجاوبوا عليهم وبلشوا يعملوا spermatogenesis after puberty. بكون من وقت ما خلق الولد للpuberty عم ينعمل secretion of FSH and LH, but they are not enough to stimulate the gonads to start spermatogenesis. Now, after puberty, the amount is enough, and they can start to respond to the FSH and LH secretions. The first step of spermatogenesis is the mitosis. Now, mitosis will lead to the formation of large number of spermatogonia that are located at the periphery. دايما منبلش من البريفري لنوصل لللومن. So spermatogonia اللي هن the spermatogonial stem cells will be proliferating at the periphery of the seminiferous tubules, and this mitosis will last about 27 days till it reaches to the cells that are called the committed cells that will start the meiosis. Now, in the mitotic proliferation, the primordial germ cells they differentiated into prospermatogonia اسمهم الثاني gonocytes. بالfetal life يعني بعده when the embryo is developing صاروا prospermatogonia بيعملوا proliferation for a short period, and then they become quiescent. Quiescent يعني they enter a mitotic arrest, يعني ما بيعودوا يعني بيخلصوا الميتوسيس وبيطلعوا بجي زيرو. ما بيعودوا بيعملوا division. They exit the cell cycle into a G0 mitotic arrest. Okay, they stop. Okay, they stop dividing. After birth, these spermatogonia develop into what we call spermatogonial stem cells. These spermatogonial stem cells عم نحكي after birth مش after puberty. After birth من الوقت ما يخلق للpuberty prospermatogonia بيتحولوا لسpermatogonial stem cells وبينتقلوا من ال center of the seminiferous cords للperiphery toward the basement membrane. بعدنا عم نقول seminiferous cords لأنه نحن بنعرف إنه بالpuberty بيصيروا seminiferous tubules. بس يوصلوا على periphery they remain quiescent يعني بيناموا لحديث البيوبرتي. بعد البيوبرتي they get activated again وببلشوا يعملوا ميتوسيس بالبيسل كومبارتمنت، بيكون صار اسمهم seminiferous tubules، بيصيروا يعملوا division وبيرجعوا بيتحولوا لسبرماتوغونيا بي، وسبرماتوغونيا بي بتبلش تعمل ميوسيس. يعني primary spermatocyte will start meiosis 1. meiosis 1 بصير في عندي secondary spermatocyte بيرجع spermatid etc till we reach to the formation of the sperms. Therefore بالأول مرحلة وقتها كان عندي primordial germ cells عطاني what we call as the spermatogonial stem cells اللي هن type AD spermatogonium. type AD spermatogonium اسمها هيك لأنه it's when we look at it under the microscope it has a dark nucleus، بينما type AP of the spermatogonia it has a pale nucleus. شو الفرق بين الاثنين؟ الأول وحدة اللي هي type AD هي انعملت من البروسpermatogonia. So prospermatogonia lead to the formation of type AD spermatogonium. Type AD spermatogonium will divide by mitosis after puberty to give more AD and AP. طب كيف one cell can divide into two different types of cells? This is possible because type AD spermatogonium can undergo what we call asymmetrical mitosis, which means it gives rise to two cells that are with two fates that are different. That are أول شيء this one cell that is AD مثلها اللي هي بتضلها stem cell، والتاني AP هي stem cell ولكن at a later level will be differentiated into type B، و type B is a committed stage بدها تكفي للآخر. So type AP عندها القدرة تتحول ل type B and this type B will lead to the formation of secondary spermatocyte and then spermatid and then sperm. بينما type AD لا، it is the reserve population، هي population اللي حتعمل لي regeneration كل الوقت ليضل عندي stock. بينما type B بنقول له progenitor type أو precursor type لأنه هو اللي حيعمل لي type B that will be the committed spermatogonia. type B is a committed spermatogonia. فإذا في عندي 3 أنواع spermatogonia، 2 هن stem cell أو spermatogonial stem cells، وحدة is committed stem cell. type A هي spermatogonial stem cells بس هي بنسميها reserve population. التايب AP هي spermatogonial stem cells ولكن بنسميها progenitor أو the precursor cell. والتايب B هي committed، it's no more a spermatogonial stem cell. This one is the committed one. I will stop here for today and will take your questions.